Next talk, we have the controlled, agile, and novel Francisco Chavez to talk about this. There, I got it, Joe.
Thank you. This is a project that Chris Scholl and Jim Bellingham and I started many years ago at Imbari when Jim was still there. He's here now with you guys at Hui. What I'm going to do here first is just introduce some of the players and some of the things that have been talked about already earlier will allow me to go through these things a little faster. Dana already mentioned our long-range AUVs, and we'll talk a little bit about those in a little more detail. And then the integration of a third-generation environmental sample processor in there. Talk a little bit about wave gliders. You heard a lot about the sail drone. There's this spray glider that we've been using. There's a project at Imbari to build coastal profiling floats that sit at the bottom of the ocean and therefore are not advective offshore and remain there. And we've been adding sediment traps with rotating cups. I won't talk much about that. Talk a little bit about the Dorado class vehicles, and Dana talked about the IMAP. I'll talk about the CTD vehicle that's working up in the surface. And again, with these devices, we can do lots of different things like pull acoustic devices to sonify the ocean. So Canon was all about bringing together different engineering efforts at Imbari and using them in process-type experiments. We hope to improve the predictive understanding on long-term and large-scale problems by developing a sustained ecosystem following and resolving observing system that leads to improved parameterization of models. Here was our dream. This, for some of you who were in the Jagoff's era, you might remember this figure. Can we follow a molecule of CO2 all the way through here or here? And that, of course, is still a dream, but now it's more of a semblance of reality than before. As Craig mentioned, if you want to follow processes in 4D, that you need a number of things, platforms, and then I'll talk a lot about instrumentations and samplers, novel analytical methods, and software. We need to make, Oscar mentioned this, we need to make these systems much smarter and be able to do things on their own. And then we have developed some tools for analysis, and I won't talk much about those at all. Here are the Imbari AUVs with samplers. The larger AUV has these gulpers, which suck water, and we can collect about 2 liters of water from them. The smaller vehicles have a small little sampler with bags on it that allow us, in this case, we can have about 20 of them, and in this case, we can have about 15 of them on the long-range AUV and use those at will. We spent a fair amount of time calibrating how well these samplers work in relation to sort of the gold standard ship-collected Niskin measurement, and this is a compilation of nitrate measurements made from the AUV, both from the gulper and the sipper. And we've done this for primary productivity and chlorophyll as well. There's a paper in LNO where we show that for this device. I'll start introducing you a little bit to the environmental sample processor. I think many of you are probably familiar with it. It's now available in a commercial version, and it's very large, but it's been used a lot for harmful algal bloom studies, and in this case, we used it on a drifting platform to look at variability in microbial activity in a paper by Otteson et al., where we followed the ESP over the course of a week. We didn't have the long-range AUVs then. We did some of the Dorado work around it. I'll show you a little bit about how the long-range AV has helped improve that. Here is a very full slide of what these long-range AVs are capable of. If you remember Oscar's slide, there was actually a little bit of a gap between the gliders and the ships. This vehicle is intended to fill that gap. It has about 14 days of endurance with rechargeable batteries, but has about a month of endurance if you use primary batteries. And I'll show you some examples of how far we can go and come with those. It has a core set of sensors. It has a wide variety of communication devices. It can actually act as a glider if we wanted to. It's got very efficient propulsion, and it can come and we can do similar things as Oscar talked about. We can extend the nose. We can make it nosier, and in this case, we've added the 3G ESP, but we can add other devices there. Here's a comparison of a section that was done out to 500 kilometers. We do this routinely. We've been doing this routinely in collaboration with Dan Rudnick. Craig showed those sections off Monterey. The LRUV is 3 times faster than the glider, not quite as fast as a ship, but certainly collects much higher resolution data. And in this case, we were able to come out supporting a cruise that was going off of here, came back, a drifter was released off of the vessel. The LRUV was able to follow it, and then it still could come back in and did some collaborative work on a boundary layer experiment inside of the bay. Here's an example of the LRUV following a drifter with the ESP autonomously, essentially. It followed this drifter over the course of 5 days, and if you put the ESP in a Lagrangian framework, you see how well we captured the environment around which the ESP was taking its samples. The ESP lab has worked very hard to miniaturize this device, and this is the new iteration. It's been tested only a couple of times. I'll show you a little bit about that. It fits in the midsection of one of these long-range AUVs. It has a series of cartridges that take seawater in and pump seawater out, and that water is filtered onto these filter systems. There's reagents in these reservoirs. There's a couple of different things that can be done with these filters. You can archive them with RNA later, or you can lyse the cells and do onboard genomic analysis. There's a new qPCR assay assay that's being developed that can be used for species-specific targets for like pseudonychia or other things, but it's also been developed for anchovies, sardines, mackerel, krill, and other important critters. In this canister here, there's also a domoic acid sensor. Of course, it had all those requirements. You don't have a lot of power on board this vehicle, so it had to be very low power. I'm going to take a little bit of a detour here and talk a little bit about eDNA, which has already been mentioned several times in this meeting. And for those of you who are not familiar with it, material for either suspended material like bacteria or phytoplankton, but other material exists on droplets of water. And theoretically, one can go from microbes to whales with a single sample from an ESP, for example. You can either do this by looking at everything through next generation sequences, or you can use these species-specific primers that I talked about, and the later is a lot easier. Just an example, and of course I picked a couple of the best examples here. Here is a comparison of a diver who had bottles of water with him or her, and was counting fish or other things as he or she made their way offshore of the Hopkins Marine Station. And in blue are the DNA estimated concentrations compared to the visual counts. And so you can see that at least in both of these cases, there is some resemblance of reality. So there is some hope that this technique can be real. The other thing, of course, is that the eDNA picked up a lot of things that the divers didn't see, like residues from cormorants, et cetera. Keeping along this little detour, I'll tell you a little bit about my office is right here at Mbari. I don't know if how many of you have been there, but we had a spectacular show of whales there over the course of 3 years. And they were all mostly concentrated in here, and they, we think, were preying on anchovies. Start here at the bottom. I probably should have put these in a different way. On a cruise that we were on, we were able to use echo sounders to identify a big blob of fish at the bottom of Monterey Canyon. The LRUV group went down almost the next day after we were there to deploy a benthic instrument. When they got down to the bottom, they couldn't see because whatever it was so thick. And when they came back up to the surface, the ROV was full of anchovies. And I was kidding them that they built the most expensive net ever. So we were able to confirm that indeed there were lots of anchovies at the bottom of the bay. And we were also able to confirm that we can use the LRUV with 3GSP to identify the presence of anchovies. So then we took historical samples that have been frozen in liquid nitrogen and put them together with these whale abundance sightings and also with the temperature anomalies. So this is a little bit of a busy graph that runs from about 210 to 216, where in blue are the temperature anomalies, in red, in blue and red are the temperature anomalies, red, cooler, blue, warmer. And gray are the whale sightings at this one location, and in green are the anchovy eDNA, and you can see there's a nice relationship between these processes. And so we've been using these process experiments to look at particular features. We had a postdoc at Mbari, Jason Smith, who made some very detailed measurements of ammonium and nitrification in the area, and found that there was a hot spot. And I think most of the and this is an area that we miss in looking at the biological pump, because it's a very difficult area to work in. It's very close to the surface, but most of the action is right in the thermocline, right below the euphotic zone. And so we wanted to look at the dynamics of that area. And so we deployed one of the long-range AUVs and had it stick with a particular isotherm. And then and that LRUV had a modem in it that communicated with this wave glider, and the wave glider could follow that vehicle as it made its way around the ocean. This other LRUV also had a modem, and it was able to follow it. So here we are trying to follow this device fully autonomously, and hopefully this animation will work. And you'll see how that happened. In green is the drifting AUV, in red is the wave glider, and in orange is this other AUV that's doing loops around it. And it did this they did this basically on their own over the course of 3 days. You'll see the orange go away. That's when the ship came in to make a measurement next to the drifting device that was drifting at the depth of approximately 40 to 50 meters. Another thing that we're doing with our vehicles is adding software to them that is intelligent. In this case, the AUV goes across a chlorophyll maximum. It figures out where it is, where that chlorophyll maximum is, and then on its return transect, it collects the sample. Here's a nice example of how that was used in a recent paper. We also, and I'll get to this in a little bit, we also have algorithms that allow us to detect fronts. And in a nice paper, recent paper by John Ryan et al., all of these samples in this harmful algal bloom episode were triggered using this algorithm and analysis of pseudonychia and domoic acid was able to be made, and as a result of that, the ability of the AUV to target these layers. Okay, Bob Sterner yesterday talked about the flood of data. This is a really nice quote by Carl Banza, which was done over 20 years ago, and we're still quite there. We're still, I've highlighted this area because that's the one I wanted you to, but we're still worrying about basically the same things that we were worrying about back then. But he says we'll live on a wired earth. Using the data so gathered, however, will demand massive improvements in scientific concepts, and this improvement is already the task of the present generation. And I think this is a really important message. Less than a couple of months ago, we had a pretty intensive experiment in Monterey Bay where we deployed for the first time, there's actually 6 long-range AUVs here, that we deployed 5 of them at the same time with 4 gliders, a wave glider, and 2 ships. This is one of the tools that we use to sort of manage our information. That period had 8 million, I can't read the exact numbers from here, more than 8.5 million data points. And so trying to figure out how to work through those, I think, is a really important message for our younger generation. And I'll tell you a little bit about this experiment next, and then I'm almost finished. As I said, earlier I showed you we were looking at this hotspot at this interface. We've also been looking at hotspots at the surface along fronts, and our fisheries colleagues are very interested in these because they can explain the general distribution of fish based on the average concentration of plankton in the surface waters. So there has to be areas where things are concentrated. We know that too because we've tagged animals, in particular sea lions, and they always tend to go and forage at these very similar locations. They tend to be very close to the self-edge and sometimes in these fingers. Anyway, this was the basic distribution of our fleet, with the gliders being further offshore. We worked collaboratively with Andy Thompson and others who had a Keck Institute funded project where they're trying to use vehicles and models in a very similar way that Oscar talked about earlier. Anyway, this is the typical sea surface temperature pattern in the spring, with colder water coming from Monterey Bay, from Año Nuevo south across Monterey Bay, with these strong fronts developing. And we use the vehicles to find the fronts for us because the most of the time it's cloudy in Monterey Bay in this time of the year. And we worked collaboratively with the Reuben Lasker, who were doing their rockfish surveys, so that we could start to build a data set of comparisons between what they were measuring in their net tows and what we could measure either with the ESP samples or with eDNA samples in general. Dave should have been up here. We had a really exciting plume and bloom there. I mean, it was huge. We kept going into this fresh water that had high chlorophyll, so we kind of knew it wasn't the California current. But it formed two nicely frontal areas that we sampled repeatedly with the, and we did this with multiple ways. We're trying to ensure that the data collection mechanism of ESP matches the data collection mechanism that we use with a standard filter collection, and that there is perhaps some relation to what's out in the environment as collected from nets. So we spent a lot of time trying to match those two systems. The frontal detections worked extremely well. They let us know exactly where we would go, and we worked collaboratively over the course of the experiment. And I'm going to end with this figure, which is near and dear to my heart. For almost 30 years, we've been running a time series at Monterey Bay. This is the record from the M1 mooring from about 1989 to the present. And you can see all kinds of interesting variations that some of our speakers in earlier days talked about. The big 97, 98 El Nino, this cool period of the Pacific Decadal Oscillation, the blob, and then the El Nino thereafter. We've collected probably the longest time series of ocean acidification in any coastal environment, starting in about 1993 to the present, by doing shipboard underway measurements. And we've now transferred that technology over to a wave glider. We could use the sail drone as well. And we're doing those transects with that device, and we're starting to transfer the shipboard measurements that we do on a routine basis to these vehicles. And so that's one of the other utilities for these platforms and instruments. And I wonder if there is a future of process time series. And what I mean here is we pick these places in the ocean where we go back to repeatedly. And in this case here, I'll use, this is right next to a frontal area. So when we go back and forth here, we create a lot of noise. Should we be sampling these features, these processes, spatially, in a spatially varying way rather than a fixed location? And I'll leave you with that thought, and we'll all sit down and take some questions.